3. Ben? Austin Kershaw.
Can I put this stuff in?
Sure. Okay, we have 2 things on the docket today. Number 1 is we need to do a little bit of follow-up from this week's lab, and then also need to give you a little bit of introduction about next week's lab, actually the next 3 weeks of lab, because this will be our last project. And then whatever time is left we can use to answer any questions you might have or to do any other review. Did you want to say something about the lab?
Yeah.
Pre-lab?
In preparation for Monday, right, and hopefully if you haven't done it already, do it then. Be sure to read over the section called the employee manual, because there's a lot of new terminology and techniques described in there that we'll be doing on Monday or next week, so be sure to read that over. There's a project overview that follows that, right? We're doing this in the guise of being employees of a pharmaceutical company, right? So the way that employee manual is written, as well as the project overview, it's assuming that you've all been hired by Grizzly Pharmaceuticals, and we have a project. And I mentioned to some of you we're going to try to clone a gene that codes for a protein that is going to be used to make a vaccine for Hanover fever. Okay? So be sure to read the project overview. In lieu of a quiz, I want you to complete preparation of recombinant plasmid. It's on pages 6 and 7. Just questions 1 through 7. You can't do 8. We will do 8 in class. Right? I will check to make sure that you have completed that. Awesome.
Thank you.
Right?
Okay. So let's do a little bit of review from the lab we just concluded this week with the...
Those of you who had lab yesterday, be sure to check your cultures today. All right? Because you won't be able to get into the lab on Saturday or Sunday. It worked very well. You have good results. So get into the lab today before 5 o'clock.
It takes, 5 minutes?
Yes.
Okay, so Piccolo follow-up. So what's ampicillin? It's an antibiotic, right? Inhibits cell walls. What happens to normal bacteria that are growing on plates of agar and ampicillin? They don't grow, right? Because the ampicillin is killing them. So the original cells that we start out with are ampicillin sensitive. Okay? They're killed by the antibiotic. Oops. So here's our plate with ampicillin and agar, and we plated out normal, non-transformed E. coli onto those media, and we don't get any colonies. We don't get any growth. What is beta-lactamase? Nominally, what can you tell me about it? It's some kind of an enzyme, right? Okay. Ampicillin and penicillin and all those other psyllin antibiotics belong to a class of antibiotics called beta-lactam antibiotics. A beta-lactamase enzyme is an enzyme that breaks down the antibiotic and destroys the antibiotic. So if a bacteria is resistant to the antibiotic, one of the common ways it develops resistance is to have this beta-lactamase enzyme. Okay? So resistant bacteria have beta-lactamase. Sensitive bacteria to the antibiotic don't. So what happens to bacterial cells that produce the beta-lactamase enzyme when you grow them in ampicillin agar? Now they grow. So we should get colonies. Okay? This is not the normal phenotype. The bacteria we start out with are beta-lactamase sensitive. Or, I'm sorry, ampicillin sensitive. They don't have the beta-lactamase enzyme. As a result of the transformation that we do to them, they acquire this particular trait. They're now resistant. So what's GFP? Green fluorescent protein. Okay, this is a protein. So this is something that is the product of gene expression. And this particular protein is of interest to us because it has a particular structure that absorbs light. And when it absorbs light, electrons are excited and we get fluorescence. So we get glowing green under ultraviolet light. In our experiment, where's the GFP gene located? The gene. It's on the plasmid, right? It is on that P-glow plasmid. So here's our little P-glow plasmid, that little piece of circular DNA. And one of the genes on this plasmid is this green fluorescent protein gene that codes for green fluorescent protein. This particular protein also has this gene called BLA. BLA stands for beta-lactamase. And this encodes the information for the enzyme that breaks down the antibiotic ampicillin. So this is the ampicillin resistance gene. And in this particular region right here, this is a promoter. This is kind of an on-off switch that regulates whether GFP is expressed or not expressed. So if arabinose is present, it interacts with this promoter and turns on the gene. If arabinose is absent, it doesn't interact with the promoter, obviously, and therefore GFP is not expressed. So how do we manipulate E. coli to take up the P-glow plasmid? What do we do to it? We heat it, we heat shock it, and we do something else. Yeah, we put it in this solution that we call transformation solution, which basically does an osmotic shock. We manipulate the cell so it's more likely to take up that P-glow DNA from the environment, because we just mix the two together, right? So we need to get that cell to uptake that particular plasmid. So heat shock and osmotic shock. Okay, we already talked about this. Arabinose is a sugar. And that sugar must be present in order for GFP to be expressed and to form the GFP protein. Okay, so here's our 4 plates. We have 2 minus DNA plates, and we have 2 plus DNA plates. What's the purpose of the minus DNA LB plate? What's that plate there for? And it is one of our controls, right? But what's it checking? What if we didn't have that plate and we didn't get growth on some of these other plates? There's a couple possibilities why we didn't get growth, right? Maybe it's because the ampicillin killed the bacteria. But maybe it's because the bacteria we were using weren't viable. So this minus DNA LB plate, this is non-transformed E. coli. We haven't added any DNA to it. This is just regular old agar. Those particular bacteria then should be able to grow on that plate. So lack of growth on this control just invalidates all your experimental results. You won't get anything. Which plate should be compared to determine if the bacteria have acquired the ampicillin resistance gene? This is our best comparison right here. We compare these two. There's only a single difference in variables, one difference. And the difference is this one doesn't get any P-glow DNA. This particular one does. Now, one of the questions that was asked on your lab hand-in, it said, on which plate or plates would we likely find transformed bacteria? Well, likely we'd find them on this plate because we mix them with the P-glow plasmid, but we could also find them on this plate. So what's this particular set of comparisons doing for us? This particular comparison isn't really best at telling us whether transformation has occurred. This particular experiment is really set up, and this comparison is set up to show the importance of arabinose in expressing that particular gene. So this is a comparison that we should make when we want to see whether arabinose is required for expression of the GFP gene. One of the other questions asked on your lab hand-in is, what is the control for this plate? This is the control for this particular plate. Why? Because they differ in one variable only. You couldn't say, for example, that this was the control for this plate because there are two differences in those particular plates. Presence or absence of DNA and presence of or absence of arabinose. So single variable difference is important. Okay? So the control plate for this is this. So what do these results tell you about bacterial transformation with the P-glow plasmid? Okay? So here we have a minus DNA LB amp. Here we've got a regular LB DNA, or LB minus DNA agar. Here we've got LB amp arabinose, and here we've got, is that plus DNA LB amp? So these are good results because we get growth on the minus DNA LB plate, so the bacteria are viable. Over here, we haven't added any DNA, so these bacteria are still sensitive, and they're being inhibited by the ampicillin. Okay? What would happen if we got some growth on here? Then our bacteria we were starting out with were resistant. They weren't sensitive. Okay? So that's an important plate to do. And then here, we've got growth on both the plus DNA plates. So these particular cells were transformed to have ampicillin resistance. And then furthermore, we get a difference under the UV light because this arabinose plate glows where this particular plate doesn't, showing that that arabinose sugar is necessary for expression of the GFP protein. Okay, so any questions about your P-glow lab? What the results should look like, what the various controls are. Want to go back for a second? Yeah.
Another way of looking at this is that our endpoint in this experiment is dependent upon the ability of the cells to grow. So we have to know for sure that they can grow. So we just put cells, bacterial cells, on the nutrient agar to make sure they can grow. Another aspect of the endpoint is we only want to study those cells that have taken up the plasmid and have antibiotic resistance. We do not want to have cells that haven't taken up the plasmid but can grow in ampicillin. So we got to make sure that our host cell, the bacteria, isn't inherently ampicillin resistant. That's what this control is for. We took the same cells we put over here, but this time we put them on the LB to which we added the amp. Right? So these two controls tell us the cells can grow, all right, in the absence of ampicillin, but that they are ampicillin resistant. And then these are the two plates where we're studying specifically the ones that have taken up the plasmid. This tells us they've taken up the plasmid. This one tells us that those that have taken up the plasmid are producing or can produce the GFP protein. All right? Experimental design, something we started last semester in Bio 134 and have done some, is really important. If you gain nothing out of these 2 semesters except learning how to properly control for an experiment, then you failed.
Slip of the tongue, you said this was resistant. These are the sensitive, ampicillin sensitive.
Yeah, we didn't want them to be resistant,
Right. Okay.
inherently resistant.
Right. Yep. Okay, so what we're going to do now is we're going to take these experiments with transformation a step further and teach you some additional techniques in the context of this scenario where you're out there working in the real world when you have a job. And it's not uncommon when you get hired for part of your onboarding purposes is for you to be given a bunch of information about your company, what they do, and particular background information you need to be able to do your job. And so that is this employee resource manual.
A big fat binder full of information that you have to sit down and read and comprehend.
Yep.
Oh my gosh, it's not online.
So our employee resource manual is pretty pared down, and it's really going to talk about 3 basic techniques that you're going to have to be comfortable with, or 3 concepts you're going to have to be comfortable with, in order to do your job to finish these next 3 labs that we're going to do for the rest of the semester. So we're going to talk about plasmids, restriction endonucleases, and then agarose gel electrophoresis. Okay, so let's start out just at the top and work our way down. Okay, so you've already been exposed to plasmids. The P-glow plasmid is what we use to transform the bacteria this past week in lab. What are plasmids? They're small extra chromosomal pieces of DNA that are found in some bacterial cells. So if we've got a prokaryotic cell, bacteria have a single circular chromosome. Their genome consists of 1 chromosome. Okay? Many bacteria, though, have this extra little piece of DNA called a plasmid, and on that plasmid can be some interesting genes. One of the genes that's oftentimes found on this particular plasmid is the gene for antibiotic resistance, that BLA gene, that beta-lactamase gene. Now, with the P-glow, people have taken a bacterial plasmid and manipulated it and inserted a piece of foreign DNA into that plasmid, the jellyfish gene. And then we use that plasmid as a vector or a transfer agent to get that jellyfish gene into the bacterial cell. Because once plasmids get into bacterial cells, when the bacterial cell replicates, they not only replicate their own DNA, but they replicate the plasmid as well. So that's why if you're successful in transformation, all of those colonies will glow, not just certain ones. Okay? So these plasmids may carry additional genes, they oftentimes do, such as antibiotic resistance genes. And very importantly, these plasmids can be genetically modified for genetic engineering experiments. So we can snip these plasmids with basically little molecular scissors and insert genes of interest into those plasmids, and then we can use those plasmids to transform other organisms. So that's one of the techniques you're going to learn how to do in lab is you're going to work with these plasmids and you're going to learn how to isolate them, to separate them from the cell, how to make cuts in them, and then how to insert foreign DNA into those particular plasmids. It's pretty powerful stuff. Okay, we're not going to be using the P-glow plasmid in the next 3 weeks. We're going to be using another plasmid called PUC18, oftentimes referred to as PUC18. Okay, so this is our plasmid that we're going to be working with. What does that plasmid have on it? Well, it has genes for ampicillin resistance. So it's got those BLA genes, beta-lactamase genes. Okay? Another thing that it's going to have on those particular, on that plasmid, is a gene for an enzyme called beta-galactosidase. So this is an enzyme. The enzyme beta-galactosidase catalyzes a reaction that takes lactose, which is a disaccharide, and breaks the bond that holds those 2 sugars together and converts lactose into 2 monosaccharides, glucose and galactose. So this enzyme breaks this particular sugar down. Now, this particular enzyme can act on other substrates as well besides lactose. And later on we're going to see that there is an artificial sugar called XGAL. And when beta-galactosidase breaks that down, it's going to form a product. Okay? And we're going to see a color change. So it's white to blue, isn't it?
It's white.
Yeah, white to blue. In the colonies of the bacteria, from white to blue. We'll get into that a little bit more later on. But it's important to understand that this enzyme, this beta-galactosidase enzyme, that's part of this PUC18 plasmid, is going to be important in our particular procedure to determine whether transformation has been occurring or not. Okay, additionally, so that's what this LAC-Z gene is right here. The LAC-Z gene codes for this enzyme beta-galactosidase. In the middle of the LAC-Z gene is this region right here called a polylinker. And this is going to be the site where the molecular scissors can snip open the plasmid, can cut the plasmid. The molecular scissors can only cut in this region right here. They can't cut here, they can't cut here, they can't cut here. We haven't talked yet about what those molecular scissors are and what they do. This particular plasmid is not particularly big. It is about 2.7 kilobases. That's 2,700 base pairs. So it's a reasonably small piece of foreign DNA. So ampicillin resistance gene, beta-galactosidase gene, and within that beta-galactosidase gene is this site where the molecular scissors can cut this particular plasmid. Well, that's not good.
Okay, when we were doing the P-glow, all right, there were two ways to know that the bacteria had the plasmid. One was antibiotic resistance, and two, that the GFP gene, which is present on the plasmid, could be expressed. This is somewhat similar in that, whoops.
Go ahead, finish and then.
Can I have the picture?
Oh, gonna let me go back. Okay.
We have the ampicillin resistance gene, but this LAC-Z gene is going to allow us to know whether or not we have an extra piece of DNA stuck in here, because the color of the colonies will change. So in addition to ampicillin resistance, we have another marker of transformation that will be of use to us, and that's because the product of this gene, LAC-Z, will turn an artificial colorless substrate from being colorless to blue if it's intact. If it's not intact, then you won't get that change. And that's another way for us to detect bacteria that have been transformed with plasmids that have a foreign piece of DNA in here.
Yeah, so we have two kind of selections. We have ampicillin resistance or not, same thing you did in the P-glow, but instead of glow or not glow, we have this white colony, blue colony color. We'll come back and talk about that more later on. I'm sure this is probably a little confusing right now. Okay, restriction endonucleases. These are enzymes. These are these molecular scissors that I've been referring to that can cut DNA. Where do we get them? Well, actually, these are enzymes produced by certain bacteria, and they're produced as a defense against virus infection. So this is part of a bacteria's defense system. If a bacterium gets infected by a virus, these enzymes can chop up the viral DNA and basically inactivate the virus. The key for these is that they cut DNA at specific base sequences. And there are different enzymes that recognize different sequences of bases. we're going to use a particular type of restriction enzyme. Called Echo R1. R refers to the fact that this is a restriction endonuclease enzyme, a molecular scissors. 1 indicates that this was the first of many restriction enzymes that was discovered. Echo refers to the fact that this comes from E. coli. So this is an enzyme from E. coli that can cut DNA at a particular location and in doing so inactivate virus. Well, the nice thing about these particular restriction enzymes is if you take DNA from 2 different sources and cut them with the same molecular scissors, the pieces of DNA have something called sticky ends. And the sticky ends can join to one another, allowing us to hook 2 pieces of DNA together from 2 different sources. So if we have 2 pieces of DNA that both have sticky ends, they can join and form a recombinant DNA. So that's how this company that made the kits we used last week in lab, Biorad, made that piglo DNA. Was they isolated the plasmid, they isolated jellyfish genes for fluorescence, they cut them both with this enzyme, the molecular scissors, and in doing so, those pieces of DNA had sticky ends that would join with one another. And then they mixed those 2 DNAs together and they formed a recombinant piece of DNA, a plasmid that had foreign DNA inserted into it. So, here's Echo R1. Here is DNA. We've got the 2 strands of DNA which are anti-parallel to one another. This particular enzyme, Echo R1, recognizes this particular sequence. AATT, is that a T or G?
It's supposed to be GAATTC. This is the restriction sites. We don't have G.
Okay, but it cuts right here.
Correct.
And that's read in the 5' to 3' direction. So it needs this sequence of bases read in the 5' to 3' direction. So here's a sequence where the DNA would cut between the G and the A. Here's another sequence later on in that strand where it would cut. Okay? The other strand is anti-parallel. So if you're reading DNA in this particular direction, here is a GAATTC. Okay, notice the complementary base pairing here. Here's another one. But because this strand is anti-parallel to this one, the cuts aren't right across from one another. They're staggered. Okay? And consequently, when you mix this DNA with Echo R1, it makes these pieces of DNA that have these overlaps on them. Those are the sticky ends. And if you then mix DNA from 2 different sources that's been cut with the same enzymes, they'll have sticky ends which will pair up with one another, and that will allow us to form recombinant DNA. So these restriction enzymes are really important in terms of cutting DNA and generating these sticky ends that can join together. Okay, the third technique is electrophoresis. Electrophoresis is a technique that separates DNA based upon differences in size. So a little piece of DNA can be separated from a big piece of DNA. Electrophoresis is accomplished by mixing up this gelatin-like material called agarose. And when you mix up agarose and pour it into a little frame, it creates this little flat sheet to which we can apply DNA. And then if we put this in a chamber that has electrodes on it, we can run an electrical current through that particular flat sheet of agarose, through that gel, and separate the DNA. There is a buffer that is put in the chamber. I'll show you the chamber in just a second. And the buffer is, what, around 7?
pH 8.
pH 8? And at pH 8, DNA has a net negative charge. Those phosphate groups have ionized and have lost their protons, and so DNA has a net negative charge because of their phosphate groups. And things that have net negative charges will be attracted by positive electrodes. So the positive electrode will pull the DNA toward it because DNA has a negative charge. Here's something real important. When we put DNA on these gels and turn on the juice, and it starts getting pulled toward the positive electrode, smaller pieces of DNA migrate through the gel faster than larger pieces of DNA. So small is quick, fast is slow. So, here's a agarose gel. Here's this gelatin-like material that's been poured in this little tray. And in the tray we have little holes, basically, called wells, where we can put samples of DNA. We then put this into a chamber that has buffer and electrical power supplied to it. And we have a positive pole down here and a negative pole here. We put the gel in the chamber and turn on the juice, and the DNA starts to be pulled toward the positive pole. Smaller molecules moving through the gel faster than shorter molecules. Now, we'll talk about this later on, but while electrophoresis is occurring, you really can't see the DNA moving. We will put little molecules in there of a dye called a tracking dye that we'll be able to see those move, but you can't see the DNA itself separating and moving. So after the experiment's done, you have to do some staining to be able to see the DNA. So this is what a stained gel would look like after electrophoresis had happened. This is the setup we're going to use called a ProCell. I know it's a little bit hard to see, but basically there's this plastic plexiglass box, and in the center of that is this little platform. And on that platform, we will put one of those gels. There are electrodes, positive and negative electrodes, that are hooked up to a power supply. And basically, we're pulling electrical current through the gel and separating the DNA based upon its size. So here's a ProCell in action. So here you can see the plexiglass box, the little platform that you put your gels on. This is the buffer chamber. There's your electrodes. And this would be gels that had been stained so you could actually see the DNA after it's separated. So each one of these little bands represents a piece of DNA of different size. Okay, I think we'll stop here right now in terms of the overview, and next week we will get more into your job assignment and what you're trying to do, accomplish. Okay? So we've got about 15 minutes, not a lot of time. Do you have questions you would like to ask about exam? Cursor.
You lost your cursor?
I lost my cursor. Try one to do that.
You just crashed.
Let this start up. No questions at all? Photosynthesis, replication, transcription. How many of you went to mentoring last night? Okay, a few of you. Well, I was going to go through a few old exam questions, but my computer is not behaving.